Hello everyone, welcome back to the show. Today my guest is Adam Finkelstein. Adam is a VSH queen breeder and he's been breeding bees since the late 80s. Together with his wife Kelly, who we met in entomology class at university, they run a business called VP Queen Bees, which is based in Ivor, South Carolina, United States. Adam Finkelstein, welcome to the show.
Well, thank you for having me. It's nice to talk about something you do all the time, and people are interested in it. It's very flattering.
Yeah. So you've been raising queens for almost 40 years. Can you tell us what first got you into it?
You know, I in preparation for talking with you, I looked up when I first started with bees, and I think it's close. It's not 40. It's like 30, 33.
Okay. Yep.
But closer. Yeah, I want to be somewhat accurate. And I had a small certified organic market garden / farm in about 2 hours south of Washington, D.C. in the late 80s. I was fortunate enough to have the land, and I really wanted to grow high quality produce and grow organically, and I got to be certified. And this was very early on. Like, we also had a CSA, and I think we were the third CSA in the United States. And I was lucky enough to meet the original CSA people who were very inspiring. But anyway, I had this market garden and worked my butt off and grew this beautiful produce, and nobody really cared about it.
Oh no.
I was, people would say, why should I buy your produce when I can get it for half or even 3/4 of the price at this grocery store? And this was way back in the day. And so I was having a little bit of some nihilistic angst out in the garden and thinking, what the heck am I doing? Why am I working so hard to make this beautiful stuff? And one restaurant in Washington, D.C. offered to buy everything that we produced, and that would have been great. And we were excited about that. But then I would have had to drive 2.5 hours to Washington, D.C. twice a week to deliver the produce, and I would have ended up being a truck driver, a teamster, and not a farmer. So I turned that down. So anyway, that's where I was, in this nice little spot in the middle of sort of Thomas Jefferson's agrarian utopia in Albemarle County, where Monticello is. And a neighbor had to move, and he said, hey, I have 2 colonies of bees and a bee suit and a book and some gloves, and I'll give them all to you for $20 because I want them to go to a good home, and I can't take them with me. And I thought, okay. I really had never contemplated having bees or keeping bees before, so there they came. And I started to learn about them and about honeybees and eusocial insects and managing honeybees and sort of all the intricacies of honeybee behavior and the way the colony lives. And it was really interesting and fascinating. And I had a partner on the organic produce farm, and she used to tease me, and she used to say, if you spent as much time on the farm as you did thinking about those bees, we would probably be making more money. And that was kind of a joke, but that led to an avid growing interest in producing bees and making honey and learning about bees. And I grabbed as many books as I could get and read them all. And I was lucky. I had a neighbor, a different neighbor, who was a commercial beekeeper. He had about 800 colonies, and he was. This was all pre-mites, of course, and he was making really nice honey. And he offered to show me. I could go around with him and I would help him, and he showed me a lot of really great stuff that I was very fortunate to view and to learn from him, and that's how I started. And then there was one other key thing: the Department of Agriculture in the state of Virginia had like several spots in their personnel for basically it was called an agricultural inspector, but mostly what you did was you inspected bee colonies, and that was to prevent American foul brood. And since American foul brood is so virulently contagious, some states in the United States have written into their constitution that the Department of Agriculture will inspect all colonies for American foul brood, and so Virginia was one of those states. So I found out that there was an opening for a bee inspector position in sort of near where I lived. So I applied for that, and with my educational background and my enthusiasm, I guess, I got that position. And so I sort of phased out growing the market stuff and was a bee inspector for 2 years, and that gave me a lot of experience in. And I think I explained this to some people. When you're a bee inspector, you want to be able to get in and out of a colony, do what you need to do, and not disturb the bees and not seem like you were ever there. And I learned that technique, and it's really, it helps you manage your bees, because you don't disturb. You're very non-invasive, and that's exactly how you want to be with looking at colonies. So that was good, but that's how I started, and I went on from there, and I learned some other stuff, and then went to graduate school and learned about entomology and honeybees and eusocial insects, and worked with a bunch of other people. But that was the beginning. That's how I started out.
Wow, that's really interesting. So, in your state, did they want to check every colony? How often?
So this was in Virginia in the 90s.
Yep.
Now, I would think in Virginia, with the way budgets are, I think they don't check every colony.
Yep.
They check commercial people, so people that are moving bees. And what they'll do is they don't check every colony. They check in a yard. They'll check 3 to 5 colonies per yard. And you
Right.
go, say a yard has 30 colonies in it. You'll start out and you'll do colony 5, and you'll do colony 15, and you'll do colony 20, and you'll do colony 30.
Mm-hmm.
And usually the rule of thumb with commercial people is, if you inspected 3 colonies and you didn't find anything, then that was it. The yard was good and you were done, and you could move on to the next yard. And then for other folks that are not commercial, I think the way it works now in Virginia and in other states in the United States is, you call and you get an inspection. You ask for an inspection, and they'll come and inspect you. Whereas it used to be much more it was mandatory, and I think it's just based on budget. To hours, people doing. They have to do a lot of other things besides bee inspectors with their position. But it depends on the state. Like, I think Florida has a much more rigorous inspection protocol, just because there are that many more bees, and there's also African bees in Florida. So inspection is imperative for safety. Um, and I would say probably like California probably has a pretty intensive inspection protocol, and then a state like North Dakota, where um, there's a lot of honey production and large operations, running thousands and thousands of colonies. They probably the state of North Dakota probably has a fairly rigorous, um, you know, each beekeeper has to show their certificate of entry, and if they don't have a certificate of entry, then they would get inspections, that kind of thing. It's sort of the inspections depend on the level of uh, sort of intensity of the agricultural production for the state. So, so in my state here in South Carolina, where I live now, I think I'm the only commercial queen producer in the state, and there are I think there are 4 or 5 migratory people with colonies over 1,000, and I rarely see the bee inspector. I mean, I talk to him every year, but he comes out every now and then, but he pretty much we have an understanding where he knows we know what we're doing, and he's busy doing other stuff. So unless I call him and say, hey, I need you to come and take some samples for me, um, he doesn't ever show up. So I think it depends on where you live.
Right. Interesting. So let's talk about something else. Let's talk a little bit about varroa sensitive hygiene. Can you tell us what got you interested in that?
Well, that's a topic filled with interesting anecdotes. When I was a bee inspector in, I think it was 92, varroa had just started to become a problem, and one of the commercial beekeepers that I was inspecting had a yard that he had moved in from Florida, and I went to inspect this yard, and it was the first time I ever saw varroa. And I was looking at colonies, and the workers had they had like 3 and 4 mites on each bee in
Oh no.
whole colony. Yeah, it was bad, and sad too. And I knew right away that this guy was going to lose all the colonies in the yard, and probably he was going to lose all his bees. And I thought, if I could do something with breeding, it would be to focus on breeding for mite resistance. That would be a worthy thing to concentrate on. One, because it would resolve this problem that I was seeing right in front of me, and two, it would be a great challenge, and everybody's always up for a challenge, but that's when I started to get interested in mite resistance, and then luckily enough, at the time, the Baton Rouge Bee Lab in Louisiana, which is a southern state in the United States, which the Baton Rouge Bee Lab focuses on bee breeding, so using hereditary mechanisms and selection to improve honeybees. They were starting to work on mite resistance, and they had two. They were very smart. They had two sort of phases of their work. One was to find a mechanism for resistance within the United States population, so a sort of an inherent, innate resistance. And then the other was to find some mechanism of mite resistance that they could import or from somewhere else, and then they could then incorporate into the United States population. So VSH came out of the first phase, which was, finding mechanisms within the bees themselves in the United States and breeding for those mechanisms, hereditary mechanisms. And that was the beginning of that was VSH behavior. And John Harbaugh at the Baton Rouge Bee Lab started that with a bee. He was a professor, I think, at Michigan. His name was Dr. Roger Hoopengarner, and he and Harbaugh started working on selection for VSH as a mechanism for controlling mites and mite growth. And then they called it, of course, SMR, which stood for suppressed mite resistance. But that was the initial VSH kickoff, and I was interested in because of course it was something that was absolutely instrumental in my goal of finding mite resistance. And Dr. Harbaugh had a policy where if you had the ability to do insemination, so you could control your crosses, he would share breeding material with you. So my wife Kelly does all of our insemination, and at the time she was not my wife, but she was, we were partners, and we contacted Dr. Harbaugh and said, we're in if you'll share some mite resistant stuff with us, and he did, and we got some of those very first queens. I think we actually got one of the first ones that was really any good. And they were, at the time, they were actually pretty bad. But I could tell you why they were bad in a different question. There's nothing wrong with what they were doing. It just what they were selecting for. They weren't selecting for commercially viable bees. They were selecting for mite-resistant bees, and there's a difference. But that's why I'm interested, or where my introduction to VSH came from. And then everything that I have and what I produce and what I work with all carries VSH, the propensity to pass along VSH behavior. And that's the only reason why we have I think we're sitting in this yard here at home where we have about 100 colonies, and none of them are treated, and none of them have been treated for mites for 23 years.
Wow, yeah, that's really interesting. That leads me nicely into the next question, actually. So, with treatment, do you think the treatment should be used in association with VSH bees?
Well, asking me about mite treatments is kind of a funny question because I don't know much about mite treatment because I don't do it. But I can extrapolate from biological knowledge, and sort of the answer that I would give is if you have bees that are not resistant to mites and they're going to die, treating them is probably better than letting them die.
Right.
Unless, of course, you are selecting for mite resistance. One of the things that it's called the bond method, the live and let die method of mite resistance selection. One of the keynotes, foundational aspects of that philosophy of selection is you have a bunch of candidates, you don't treat them, and if they manage to survive, they're obviously something is keeping them alive that has to do with mite resistance, so you propagate them and see what happens. So in that regard, not treating is great, and we have a whole selection protocol that we use in our breeding project where there are periods where there's no treatment for a specific amount of time, and then mites are counted measured and counted, and then the colonies with the lowest mites with no treatment are used for the next phase. But a regular non-breeding individual who has bees should most definitely, um, keep a keep a handle on their colony's mite level, and when the mite level gets to a specific threshold, they would then treat. And that's been a kind of a sticky question. No one has really wanted to commit to what a threshold level would be. And mites in general are very hard to nail down because the mite population changes over the season. So at some levels of testing, some colonies testing levels, the colonies will be really low with mites, and then at other levels, other times they'll be, they'll have a certain number of mites, and it will seem like it's low, but it's actually not. It represent and a representation of the level of mites in the colony. So I think what people say, who deal with treatments, and extension people that I've heard, is that you monitor your bees, and you should monitor them 2 or 3 times a year. Um, Monitor them in the spring, monitor them after the honey flow, and monitor them as the bees are winding down in the fall. And at any point in time, if you're finding, um, 5 or 6 to 10 mites per 100 bees, and there are various ways you can do that test. But if you're finding that level, then you should probably treat them. Um, and another thing that I've heard is that people mix up the treatment. So one year they'll use one treatment, and another year they'll use a different treatment, and that works pretty well. So
Right. And so with VSH, is it possible to have hives that never have mites ever?
um, I don't know. That's a good question. I think that's, um, probably not realistic, just because of the way mites work and the way bees work. In fact, if I'm looking at a hypothetical group of colonies and I test them for a mite level, and I'm finding 0.00000 mites per 100 bees, I'm concerned that my tests are off or something's going on, because that's just not what you find usually. Usually you'll find 1 mite per 100 bees. You'll find 5 mites per 100 bees. You'll find 8 mites per 100 bees. You'll find a lot of 2 and 3 mites per 100 bees. Um, but not having any mites, um, we have a bunch of colonies that test zero too, so it's 0 mites per 100 bees. But so and then over the course of the season, they're 0 and 0. But it's just they're the mites are going to be there. They're very well suited to, and they have a adaptational, a niche within the eusocial colony of bees. So if they can figure out a way to live within the bee colony and not kill the bees, they're going to be there. And I don't think having mites is a low level of mites is very bad. I think that when you have when the mite level gets to be extremely high, that's when there are issues. But I think the concept of 0 mites, is not really a biological way to think about things. And look, one is dealing with an animal. You're dealing with a natural, in the wild part of nature that's multicellular and has all kinds of fascinating and intricate functions. It's not perfect. It's not going to fit your concept of this is a great colony because it has no mites. I mean, that's in a way sort of, not realistic. And what happens with people and bees, and I've seen this, and I kind of make not make fun of it, but I have a good time with it. Um, Bees are bees, and they're going to do their honeybee. Um, they're going to live their honeybee lives, and we're mammals, and we're humans, and we live our human lives, and we have certain expectations of our bees that very often are not realistic. um, the classic thing I love when people say, well, started out a colony and it grew, but it only grew from the middle of the hive to one side, like it didn't fill in the frames of the other side, and I had to move the frames around and do everything and all this. And I always think to myself, I got a bunch of colonies that do that. They pick one side and they go up vertically on one side. We call them sidewinders. And I don't know why they do that, but I'm not going to worry about their symmetry, whether they're going to fill both sides of the box up. If they're going to fill one side of the box up and they're doing well and they're healthy, then so be it. So my sort of point with the zero mite, or my response to the zero mite question is, I don't know. I'm sure there's some colonies that have zero mites, but I don't think having a few mites is bad.
Okay. So, based on photos I've seen of your bees, it looks as though you use exclusively YSP 10 frames with no excluders. Why have you chosen this setup for breeding your queens?
I think that's a hilarious question because it really has nothing to do with breeding bees.
Okay.
But I'll answer it. Whatever the term you're using for the box size is new to me. I've not heard that. That sounds like an Australian term. The United States calls it a medium or Illinois depth. It's 6.25, I think, is the frame size, and I use those because they're pretty much all purpose. I use them for brood. I use them for honey. If bees need to put honey somewhere, they'll use those frames. And I use one box, and that way I only need to buy one kind of box, one kind of frame, and everything is the same. And that's a decision I made a long time ago, and I'm very happy I made it. I have my nukes have those same frames, and I make my nukes out of those same boxes and divide them into doubles. But that's what we standardized on, and that's what works. I'm not a honey producer. I'm not worrying about making lots and lots of honey. My bees do make a lot of honey, and in fact, at times it gets in the way because I really need those frames to be empty so I can get brood in them and not have them full of honey. I don't have an extractor. I decided to get rid of an extractor years ago just because it was in the way. But that's what we use. That's a good. It's not a shallow and it's not a deep. It's an in between frame, and it works pretty well for all purposes. I usually, going into the winter, I'll have the bees will be in the two boxes, and then there'll be a solid box of honey above those two boxes. And then by February, the bees will have moved up into the second and top box, and the bottom box is completely empty, and you just then take the bottom box away, or else you flip the bottom box up onto the top, and that gives them something to move into. And that's how we've done it for a really long time. Queen excluders, sometimes I use them, sometimes I don't. I use them to exclude queens, like if I'm trying to make something queenless, or if I don't know where a queen is in a colony where I need to have queenlessness. I'll throw a queen excluder in there, but I don't necessarily use them for honey production or not honey production. It's just more of a queen excluder is more of exactly that. It's just for excluding the queen to find out where the queen is. If I was making honey, I probably would use queen excluders because they keep the brood nests segregated from the where the honey is stored, and it's a big waste of time if you have one of those 1 in 40 queens that likes to go up and lay brood in the honey area. You've got to separate all that stuff out, and it takes a lot of wastes a lot of time. So, queen excluder, I would use them if I was a honey producer.
Okay. So you offer breeders in Italian, Carniolan, and Poline 2.2. Can you tell us a little bit about these strains?
Sure. First of all, almost everything that's used to describe strains of bees is ancient history. Um, Genomically, so if you look at the base sequence of the mitochondrial DNA in bees, there are certainly Italian, quote unquote, Carniolan, quote unquote, and whatever. Another good one is Apis mellifera mellifera, which is the English bee. There are markers that show that bees come from those matrilines, but the way we have grown agriculturally, um, almost all the unique traits that segregated those strains back in the late part of the 19th century and the early part of the 20th century, that stuff has all been mixed up and mixed together. So, for example, people say they want an Italian queen. I assume that means they want a queen that's very productive, lays a lot of brood, turns most of the nectar that comes in into brood, draws out comb really well, is gentle, and is very fecund. Will make a lot of brood and also a lot of honey. Whereas if someone asks me for a Carniolan, I'm thinking they're going to want a bee that is somewhat more frugal with how they use their incoming resources. They tend to put a lot of the resources away, store it, and they wrap the brood nest in it, and they tend to not lay as much brood unless the weather is really good. And then when they do go to town and really start to bring it in, they really bring it in all at once, all of a sudden. And that's, those are the classic traits of Carniolan mountain bees that were selected over the years by various generations of beekeepers, because those bees, you didn't want them to start up early because it would possibly get cold. So you wanted them to wait and sit in their hives or skeps or whatever they were being contained in until the weather broke, and then you wanted them to take off. Whereas the Italian bees were kept in an area that had a much milder winter, so having them be more productive more quickly and in a way more sensitively based on what was coming in was better for that warmer, sort of more forgiving climate. So now when I have on my website Italian, Carniolan, and Pauline 2.2, I can tell you that the Carniolans are from stock we have that we select for the Carniolan traits, and every now and then I will get some genetic material, semen from one of probably the only person in the North America that really has a good curative, I guess if she would be a curator of the germplasm for Carniolan. I'll get some stuff from her and make some crosses and see how they do, and then possibly select those back into what I've already got. I do that every 4 or 5 years. The Italians are basically what I have as Italian, quote unquote, is the stuff that I have that's mite resistant, that is light in color, makes a lot of brood, is really, has a sunny disposition, is very optimistic. Um, that's what I call my Italian, but it's usually the hallmark is it's light and broody. And the Pauline 2.2 is pretty much our breed that we've come up with and worked on for a long time. And it's just consistently productive, makes a lot of brood, is gentle, is mite resistant, and is pleasant to work. Um, doesn't isn't runny, but at the same time, you know, doesn't sit around either. It's just a good all-purpose, all-around bee that a hobbyist could use and a commercial person, commercial operator would also be happy with. So that's how I sort of would describe the strains. Um, I'll have people tell me that they have Italian bees or they have Carniolan bees, or I have even have some people tell me they have Pauline 2.2 bees, and I never did business with them. And I always think that's kind of funny because it's like, oh, really? That's great.
So, how long have you been offering that line, the Pole Line 2.2?
Since I think 2015 or 16, that's when we started that project after the USDA finished their sort of a demonstration project of taking VSH queens and producing cells from those queens and then mating those cells in a commercial apiaries drone cloud. And the concept there was VSH and mite resistance, and even back to the early SMR stuff. That stuff was selected to be mite resistant, and it was very mite resistant, but it was not selected to be productive or to produce honey or to make good brood or to pull out comb uniformly or to be gentle. So the idea was, okay, we've got this mechanism for mite resistance. Let's now take the mechanism for mite resistance and let's try to breed with that and impart that mite resistant ability into commercial bees that are good with everything else. So that was what the Pauline, basically in summary, that's what the Pauline project that the USDA ran from, I think, 2008 or 9 up until 2013, 14. That's what they did. And we went to them and had a meeting and talked with all the scientists and some of the cooperators, and we offered to continue doing that kind of selection because we think it's a good selection method, and they were all for it and supported us. And so that's where we named it 2.2 just so that we could discern that it was not the USDA's work but someone else's effort, not to make things confusing, as I know names of bees tend to get crazy and confusing. There are so many different, you can buy this, you can buy that, and it's hard to tell what is what. But Pauline 2.2 is just, we're taking candidates that have been screened for production, that are good candidates, and then we don't treat them for a period of time. And then after that period of time, we count the number of phoretic, so the number of mites on the bees within the colony. And the ones that pass all the tests, that are productive and that have low mite counts, we then use those as colonies that will cross with other good colonies. And it's very simple. It just is a lot of legwork and it takes a lot of time and effort because you have to test a lot of colonies. But it's not rocket science. It's not any fancy stuff in a lab. It's just good old fashioned animal husbandry, and it works really well. Our results show that, and we're happy with them, and our customers are too.
Mm-hmm. Excellent. So you're maintaining these lines through artificial insemination, and are you still offering classes on this at the moment?
We do. We had pretty much had a fairly regular class for multiple people set up in June every year, and when COVID hit, that ended that. And then after a couple of years, we started to get people. We were giving more private one-on-one. Someone would come and they would visit us for 2 days and get trained. Um, but I think we could certainly ramp back up into having multiple people here for 2 days. It's nice to have more than one person because then the students can talk amongst themselves and share their experiences and their perceptions. And I think it's a more enriched environment than just a single person.
Hmm. Excellent. So, can you recommend any books on queen breeding?
Okay, so let me ask you a question with that question. Are you talking about queen rearing or are you talking about queen breeding? Because they're two very different things.
I think queen rearing, there's quite a bit of information out there. I really want to know the queen breeding side of it. Are there any books that you would recommend for helping a queen breeder?
Okay, I can give you a couple books. The unfortunate truth is that breeding in the the art and science of artificial selection, under the category of biology and agriculture, is not an exact science. There's certainly has been a huge move forward with a lot of breeding in agriculture for animals that are high dollar. I mean, like dairy and meat and wool and all the fancy um, sort of mainstream cash uh, crops. Unfortunately, with bees, and everybody who is a beekeeper learns this sooner or later, where the bee people, the apicultural aficionados are always at the bottom of the chain with everything. Um, you know, you can count, you know, hundreds of commercials that mention save the bees or for you bees and this bee and that bee, but they're not those people that are making money from the commercials are certainly not contributing to anything to do with apiculture. There's so much basic research in apiculture that would be helpful to have conducted, and yet it's not. Like virgin queen physiology or virgin queen behavior. Like nobody knows anything about that, or artificial insemination stuff. I mean, we see things when we do an artificial insemination that no one talks about in research, just because no one's done it, and it's not. You can't just pick up and do research. You have to be in a research program. So anyway, with that slight rant, I will say that one of the most valuable books for heredity in an apicultural application, so learning about basic Mendelian inheritance with bees, is Brother Adam's Beekeeping at Buckfast Abbey, because Brother Adam explains how he used Mendelian genetics and his selection for producing the Buckfast bee. And that's basically, it's really interesting. And just it's great to read Brother Adam stuff because he's a monk, and he's talking about stuff that he really knows a lot about because that's all he does. And so it's very satisfying. I find reading Brother Adam very satisfying because he is truly an expert, or was truly an expert. But that's Mendelian genetics. And what happens with breeding and genetics is people learn a little bit about Mendelian genetics, which is sort of the simplest, oversimplified version of heredity, and then they think that every aspect of heredity is based on Mendelian genetics. Mendelian genetics, the example is dominance and recessive. Blue eyes, the heredity of blue eyes and brown eyes in humans. So, if you have blue eyes, you're going to have a recessive genotype, which is for blue eyes. If you have brown eyes, you could have a dominant genotype, which is all brown, or you could have a heterozygous, half brown, half blue, but the brown overrules the blue, so you're going to have brown eyes. That sort of relationship, that's all Mendelian stuff. But Beekeeping at Buckfast Abbey covers all of that. There's a second book that he wrote, which is called, I think it's Breeding the Buckfast Bee, but don't...
Breeding the Honeybee, it's called.
Breeding the Honeybee.
Interesting coincidence, that same name of this podcast, actually.
Yeah.
It's a great book, yeah.
That book is a good book. I find that there's actually more actual applicable bee breeding information in the first book than in the second book. But they're both good books. There's another book. Let me grab it so I can give you the exact title. Hang on a second. All right. This is probably the best all-around bee breeding book. And I think it was out of print for a while, but I'm pretty sure it's back in. It's by Friedrich Rutner. He was a German apicultural expert, entomology professor. And the title is Breeding Techniques and Selection for Breeding the Honeybee. And it was by him. It was translated by Ashley and Eric Milner, and it was published, I don't know if it's still published by them, but the copy I have is published by the British Isles Bee Breeders Association. And this is a really good sort of, I would say it gives you both queen rearing in the context of breeding, and then also some selection and some pretty serious foundational concepts when one is breeding. And I heartily recommend this book. Sadly, a lot of the references in this book are to breeding carniolan bees, and I think that a lot of the stuff is old. It doesn't exist anymore. You know, the breeders that are referenced have passed on, and I don't know if a lot of this stuff is actually current, but certainly the information that this book gives is very good. And then probably the most helpful thing with breeding in bees, but breeding in anything, is to look up a basic college genetics textbook. And that's handy because you can learn a lot of sort of the mechanics of heredity, and then you understand that, and then with that you can sort of apply that back to breeding any animal. And of course then honeybees are in there. But what's hard about breeding honeybees is the bees are haplodiploid, so there isn't really a male and female bee. You know, it's all female. It's just the drones, the male expression of the organism still has the genotype of the queen that the drone is from. So there's not really sexual reproduction in the sense of two different parents. It's pretty much all from one side, and that makes for some fuzzy and sticky issues when breeding. And the classic one is that if you become too homozygous, so if what you're breeding becomes too similar, you can have this devastating crash of vigor in your stock. And it's very frustrating because when one breeds, one is taking the chaos away and making things ordered and focused. That's what selection is. You're picking what is good and you're throwing away everything else. And what happens when you do that with bees is you're you're getting what you're heading closer to the ideal, but you're also getting dangerously close to becoming ruinously homogenous and losing everything you have. So we always say, it's like 3 steps forward and 2 steps back when you're breeding with bees. You get close to what you want, and then you've got to keep that on the side, and then you've got to have something else going so that you're not going to get too focused into one thing. And that can happen very fast, especially with artificial insemination. If you don't have enough diversity, you can end up with everything being very genetically similar, and you'll have half a brood pattern. Let me give you an example of this. So the 2 places that I get some of my genetic material in terms of queens, so the candidates that I use for my breeding colonies, there are 2 operations, commercial operations. One has around 11,000 colonies, and the other has around 35,000 colonies. And I work with both of those guys. Well, I have the luxury of having a pool of that size. So I'm already way ahead in terms of breeding than a lot of people, just because I have the ability to have that large sort of chance of getting something good. If you take those numbers, 11,000 and 35,000, and you look the other way, and you look at someone who's breeding their breeding bees and they've got, say, 500 colonies, I mean, it's almost not an order of magnitude, but it's a very large difference between 500 colonies and 46,000 colonies. And that's what happens to a lot of people, and they get discouraged, is that they breed their bees and they make their crosses and they keep really good records and they do all this stuff, and it doesn't really matter because they're just getting too homogenous too quickly, and they don't have a way to bring in new stuff that's also really good. So the answer to that, and a lot of people don't like the answer because it's something that humans are allergic to, is I'm being a little cynical here, is cooperation. The best way to breed good bees is to work with a bunch of people, to have a group, to have a consortium, to have a club, to have an association. And everybody figures out what they want and what the common goals of the group are, and then instead of one person having to shoulder the population in the selection, it can be distributed over a bunch of people and even a bunch of geographies, and that would be even better. And that way you're fighting against the homogeneity that is your enemy as a bee breeder. But that also involves, someone who, or a manager, or several managers, and communication and time and all that good stuff that is the enemy of anything that requires excellence. Is you have to give up something to get excellence. And with bee breeding, I love when I have a customer and I have their queen ready, and I send them an email. A lot of my communication is through email. It's just faster. I send them an email and I say, your breeder's ready, or your breeders are ready, and I can ship them next week. They can arrive Tuesday, Wednesday, or Thursday. And I'll get an email back from people, and they'll say, oh, well, I'm going on vacation, or I've got this thing at work, or blah, 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 blah. And can we work at a different time? And I always think, okay, this person is not a breeder, because they're not prioritizing the breeding material to be the number one thing on their plate. And unfortunately, since breeding takes so long and it's so involved, and requires so much attention to detail. You have to put it up front. And, you know, I wish it could be. Wish it could be easier. I wish it would be a simpler process. But to
Yeah, and
really
the timings, that you have to
exactly,
stick to them. They're the number one priority. It's tough, isn't it?
yeah. So it's that's sort of with these books. That's the sort of the wrapping that all together. I mean all the people say the same thing. You want to make sure you're seeing exactly what is there. Like the classic thing is, oh, I have these two really good colonies, and they're in this one bee yard, and one colony is at one side of the bee yard, and the other colony is at the other side of the bee yard, and then you say, oh, and you have all the bees are in a stand, all in a straight line, right? And they say, yeah, and of course that's because all the bees have drifted down to the two end colonies, so that's why they're so good. And that's you're not gonna make a good selection that way. You're gonna just pick the ones that are inflated artificially. So you have to make sure how you're selecting and what you're gauging with is accurate and not just sort of what you want to see. A lot of times also too with culling, with getting rid of your undesirable candidates, you have to kill them, or you have to give them away, or you have to take them out of your breeding program. And I know people, I do it too. It's like you'll have a queen and you'll just like the queen, and she's not very good. She's not worthy, but you don't want to make her go away. So what do? Oh, I have this nice queen. Yeah. Well, if you're a really good breeder, you get her so that she's not making any drones in an area where you're mating, or you give her to a friend, or donate her to something like that. But you got to get rid of anything that's not worthy, and you've got to find what's worthy, and that's a constant cycle.
Well, that's really good advice, Adam. I also really like the advice about collaboration. I think that's a very important one. So thanks for that.
Sure.
I think I'm aware of your time, and thanks so much. So can you tell us where we can find you, website, social media, and then we'll end it there, and you can get back to your busy day.
Okay. Again, thank you for being interested. And you all in Australia are going to have some fun with varroa. Just realize that it can be done. You can select for bees that are pretty darn resistant, but it's a whole different way to manage bees. Or you manage bees differently, and there's more testing, and it's less fun. But the naysayers who will say that you can't, bees don't have resistance to varroa, that's just baloney. They certainly do. There are plenty of resistant bees out there. It requires work to get to them, or get them. We have a website, vpqueenbees.com, and that kind of lists what we do in a sort of a abbreviated form, and it has our contact information. And I don't really I'm not on much social media. Every now and then I'll put something on Bee Source, because that was the old sort of original forum for bee people way back when. So
still very good reference.
Yeah, I'll sometimes post some stuff on there, but so if anybody has a question, they can certainly send me an email. And sometimes it takes a while when we're really busy. It takes a while to get back. Sometimes I write people back 2 months later, and then I never hear from them, and I think, oh, they don't want to talk to me. I took too long. But
they solved the problem.
Yeah, but when you're
yeah, I know what it's like. Start of spring, very busy.
Making the orders and stuff, it's you don't have enough time in the day. But that's we're out and around, and if anybody is ever in the United States and they want to, they feel like having a visit, just get a hold of us. We're pretty accommodating. We just have to schedule it so we can, have enough time. That's the main thing with us, is just putting it on the schedule. Once it's on the schedule, it's no big deal. So
excellent. Well, thanks so much for being on the show today, Adam.
You're welcome. I appreciate you.
Okay, well, I hope you enjoyed that episode, having a chat with Adam. Really interesting stories, really interesting information. How interesting is it that he doesn't own an extractor? He's solely queen breeding, and he doesn't extract honey at all. Also, he hasn't treated for 23 years. So it goes to show that it is possible to live with varroa and not have it affect your bees too badly. Very, very interesting. I really like the point that he made about collaboration. That was fantastic. If you want to get in touch with Adam, you can by going to his website at vpqueenbees.com. If you want to get in touch with me, you can at nixonbees.com.au. And until next time, thanks so much for tuning in.